every single cell has the exact same genome and the exact same genes in it. And so you have to say, to be a liver cell, turn on all of the genes that make all the proteins to be a liver cell. And that's very different than if you want to make a toe cell or an ear cell or an eye cell. And you have to turn on different genes. And so even though the genes are pretty much the same, how you can turn them on and off changes what cells you get. We're really interested in the non-coding genome, and so you might be familiar with genes, and those are really the building blocks that make up the cells of your body and make you you, but they actually only account for around 1 to 2% of your entire genome. The other parts of your genome, these non-coding regions, actually help to turn on these genes and help distinguish things from, say, maybe make a skin cell versus a liver cell. Turning different genes off and on is what helps distinguish this cell to be cell 1 and this cell to be cell 2. We see that a lot of the differences between humans that account for diseases are actually in these non-coding regions rather than the genes themselves. But the regulatory code of the genome is really complex. And so a lot of my work is figuring out how that regulatory code relates to function. So it's really just being able to take that non-coding part of the genome and learn how to read it. A lot of the work we do is being able to actually test the functions of regulatory elements. One of the techniques is called the massively parallel reporter assay. And so we can actually print out pieces of DNA, and we can encode whether the DNA has a mutation in it, hook it up to a gene, and ask, does it actually control gene expression? And so we can do this in one single tube in one single experiment. We can test hundreds of thousands of these regulatory elements at the same time. The other thing that we do a lot of work in is using CRISPR-based screens to perturb parts of the genome. We design a short guide RNA, and that pushes the CRISPR machinery to one specific part of the genome. And what it actually does is goes into the genome and it actually can turn off that regulatory region. And when we turn it off, we can ask what genes are impacted. So we're getting tons of raw data from all of these experiments, and they're all asking really fundamental questions of, this sequence yields this level of expression. And so from that, that's a perfect setup of a training data set to build machine learning models on. And so we've been saying, this code is there to be broken, and we haven't been able to do it yet. And so we take a lot of the data from our experiments, put them into machine learning models, and ask, can you actually understand how the sequence relates to gene expression? Understanding the regulatory code allows you to start interpreting how variants work in people. If you get someone's genome and it has a variant or a mutation in it, we can put it through this code and say, what might it affect? Which of these mutations that you have are actually important? Which ones are just happenstance and don't do anything? And that'll be really important as we think about things like precision medicine, where more and more people's genomes are being sequenced. Being able to really pinpoint which of those 3 million mutations you have might be important, and actually be able to triage them and think about which ones are related to your phenotypes or different diseases that you specifically might have. If you can start to read out the logic of the genome, one of the best tests for if you're a good reader is, can you actually write something? And so one of the things we've been working on is using generative AI to actually build completely new regulatory regions that have the functions we want, so we can encode functions into them. And so we've been trying to engineer these regulatory elements to be able to turn genetic drugs on just in the target cell types we want. So we're repurposing this logic of the genome to control when and where genes are on and off. Say if we can actually encode functions we want, we can control exactly where those are for a therapeutic purpose.